Welcome, everybody. On behalf of the Immune Deficiency Foundation, welcome to tonight's forum, Long COVID in the PI Community. I'm Kathy Antela, Vice President of Education at IDF. Thank you for joining us tonight. And as always, this forum is being recorded. Tomorrow, you will receive an email from IDF. It will include a link to a short survey which we hope you will complete in order to let us know how we did tonight, and also to provide suggestions for future forum topics. As always, a disclaimer. Please remember that the information presented during this event is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of a physician or other qualified health provider with questions concerning a medical condition. Please never disregard professional medical advice or delay seeking it based on information presented during this event. For over 40 years, the Immune Deficiency Foundation has served as the trusted organization for the primary immunodeficiency community, with the mission of improving the diagnosis, treatment, and quality of life of people affected by primary immunodeficiency through fostering a community empowered by advocacy, education, and research. We value our role as a patient advocacy organization. As a result of our mission, IDF seeks to ensure that everyone in the US affected by PI has a fully informed understanding of the PI diagnosis that affects them, all available treatment options, the expected standard of care, and all of their opportunities for connection within the PI community. Well, we have these wonderful sponsors who make these events possible, and we cannot thank them enough. So this year's 2022 sponsors are CSL Behring, Grifols, Takeda, Horizon, Accredo, Farming, ADMA Biologics, AstraZeneca, Bioproducts Laboratory, Kedrion Biopharma, Enzavant, X4 Pharmaceuticals, and Chiesi. Thank you so very much for your support. And we're going to hear from one of our sponsors right now. So I would like to invite Michelle Rhee from X4 Pharmaceuticals to unmute herself and share her camera and share some information about services her company provides to the PI community.
Thank you so much, Kathy. Thank you, IDF, for this fantastic educational forum. My name is Michelle Rhee, and I am a patient advocate at X4 Pharmaceuticals. Our mission is to develop treatments that have a clear and profound impact for people with rare diseases, including primary immunodeficiencies. And I'm incredibly lucky in the work that I do because my job is to help incorporate the patient perspective into the work that we do in pursuit of our mission. And so although the patient perspective isn't our only data point, it is a very important data point in every decision that we make. And so on the next slide, one of the things that we talk about is that the way that we do our business, because we are a business, is that we're a for-profit standalone research development and commercialization business, but we're a patient-focused company because we put patients first. And one of the things that we're incredibly grateful to the Immune Deficiency Foundation for, and why we've been so grateful for the partnership with, is that we're developing therapies for a couple of communities who really don't have a lot of resources. I think a lot of you know this and have experienced this. We're developing therapies for people with WHIM syndrome and chronic neutropenia. We have and when, for example, like people with limb syndrome, there isn't a disease-specific patient advocacy group, and the IDF has provided a home and resources and support that they didn't have before. And as someone with a rare disease myself, and my disease doesn't have a patient advocacy group, it's just seeing what the IDF provides and offers to the patient community is just really remarkable. Like the infrastructure and support and sophistication is something that we're incredibly grateful to be able to support and be a part of. And so we're really excited to support events like this tonight and to be able to just be a part of the community as well. So one area that we definitely, and again, this is like my personal experience where I'm like, this is very important, is within the shortening that diagnostic odyssey. It took me 21 years to be diagnosed with my rare disease. And and I know the IDF is really focused in that area. And an area that we've been able to work on also is we're providing a no-cost genetic test for anyone who thinks they might have neutropenia, basically. And so like it's an awesome program. It's one of the things that I'm most excited about. So if you want to learn more, you can contact patient info at X4 Pharma, or there's a website, but there's also information on the IDF website about genetic testing. You can kind of like go into the search bar, but it's just, as you all know, IDF is amazing, an amazing partner and an amazing support. And we're just really grateful that they are willing to let us be a part of it too. So I'll let you all enjoy and learn. And thank you so much for giving us the opportunity to speak.
Thank you. Thank you so much, Michelle. And between the presentation tonight and the Q&A, we will be hearing from some of our other sponsors via some wonderful videos that they have provided to us. So now at this time, I would like to get the show on the road. The reason you're all here is to hear this wonderful presentation that we've all been waiting for. So now I would like to introduce our presenter, Dr. James Heath. Dr. Heath is president and professor at Institute for Systems Biology in Seattle. He also has the position of professor of molecular and medical pharmacology at UCLA and has directed the National Cancer Institute funded by NSB Cancer Center since 2005. Dr. Heath received his PhD in 1988 from Rice University, where he was the principal graduate involved in work that resulted in 3 senior members of his group receiving the Nobel Prize in Chemistry in 1996. Additionally, Forbes magazine named Dr. Heath as one of the top 7 innovators in the world. So now, please join me in welcoming Dr. Heath. Thank you for being with us.
Thank you so much. So I'm going to talk to you this evening about work that we did on trying to uncover the origins of long COVID. There's another term for it is post-acute sequelae, a very technical term, from COVID-19, meaning patients who have recovered from the acute disease but have symptoms that may linger for months or, for all we know, perhaps even years following. This has been in the news a lot. I think many people are familiar with it, but these symptoms can range from neurologic, they can be respiratory, they can be cardiac, they can, fatigue, difficulty exercising, etc. And I think something that we've been learning and that I think will become perhaps hopefully apparent in my talk is that long COVID and the causes associated with it may not be that crazy different from some other chronic conditions such as post-acute Lyme, chemo brain, other autoimmune conditions that that many folks are familiar with, but on the surface may seem somewhat unrelated. And so the study we did basically, we began looking at patients at the very start of the pandemic, back in March of 2020, seems like a decade ago. And at the time, no one was thinking about long COVID. But when we got our patients to consent into our study, we wanted to follow them not just through acute disease, but out through recovery as well. And as long COVID became more and more reported, we realized that we had the opportunity to try to understand a little bit of the nature of long COVID. And so I'm going to talk to you today about that. And I'm going to, there'll be a lot of data that I'll show you. I want to give you the sort of scale of what it takes to understand the nature of a complicated chronic disease like long COVID. And so what we did basically is we did a huge number of measurements, and I'll show you. You don't necessarily need to understand the measurements, but I think it's important to appreciate the depth at which we have looked at these patients, which is absolutely critical for trying to understand something as heterogeneous and in many ways is vaguely defined as long COVID. It's almost as if every patient looks a little bit different. And then we wanted to also identify what we think are risk factors. Some of these risk factors may actually be the causal factors also for long COVID, and connect this matrix of measurements and the risk factors to the types of symptoms that patients are reporting. And we wanted to understand not only what are those past factors, those risk factors for long COVID, but how independent are they and how early in the disease course can you identify them? And a surprising thing that we found is that pretty much at diagnosis, we could identify whether a patient would or would not develop long COVID symptoms months out. And this was a finding that was highlighted in a lot of popular press, but it's the detailed study is in this Cell paper that was just published in press early in March. So this gives you a feeling for the depth of the types of experiments that we did. So we looked at serial blood draws, one right at diagnosis, which is very important, one at the peak stage of disease, and one out 2 to 3 months later. And then on each of these patients, we did a comprehensive extraction from their electronic health records to understand their histories, as well as questionnaires that we gave the patients. So that is more or less epidemiology. We did a very thorough analysis of what we could identify from the blood. And this is many things that we did. I would say we measured almost 1 gigabyte of information per patient, looking at proteins, at the circulating immune cells, the B cells and T cells, antibodies, autoantibodies, viral load, et cetera. And then a big task here is to integrate all that computationally into a model of what the patients look like. So I think when I'll use a broad term called immunophenotyping, which really means that we're trying to sample the patient's immune system in as many different ways as possible through the blood, it can be a little confusing. And so I have these symbols for proteomics or metabolites that we measure, blood measurements of viral signatures. This is single cell data that we collected on individual T cells and B cells from the patients, the electronic health records. I'll pull up these symbols as I talk about what each one of these taught us, and as I go through the data. And I'll try to give you a sort of a layperson description of what these conclusions mean. Okay, so in terms of our patients, we had about, in our primary cohort, about 210 or so patients, plus another cohort of 100 patients that we got from Helen Chu's program at UW. These patients spanned the range of disease severity. So we had some that were just outpatients the entire time. Some of the patients were basically in the hospital and on respirators. If you look at the symptoms that the major symptoms that our patients reported, they pretty much look like, if you just did an epidemiological study across the United States, they look pretty similar, and that fatigue is a dominant one. Gastrointestinal issues are less common, and so on and so forth. Patients that had severe disease are more likely to have long COVID, but not a lot more likely. It turns out that patients with mild infections also have a high probability of developing long COVID. When we look at the patients that have, after they basically recovered from the symptoms from disease, so this is now 2 to 3 months out, what this data is saying is that the patients have the signatures of having developed immunity against COVID. They have the antibodies that are protective. And in general, if you had more severe disease, you have more antibodies. If you had milder disease, you had fewer antibodies. This is something that we see in our patients. It's what's seen throughout the literature, so it's well known. And we also can tell that these antibodies truly are protective, and we do what are called cell-based assays to determine that. And that's what this data represents here in this blue dotted plot here. Okay, so what kind of preconditions anticipate long COVID? Here I've looked at a number of factors that we can pull out of the electronic health record. The dominant one that we see is type 2 diabetes. So something you obviously know about if you have before you are infected with COVID-19. It's a major factor to anticipate whether or not you will develop long COVID. There are a few others like coronary artery disease, but I don't think we have a major statistical representation of patients in our study to know how important these are. When you see stars here, that means that statistically it matters. But diabetes is the one that we really had probably 50 or so patients with diabetes. And we could say with some certainty that type 2 diabetes is going to lead to long COVID symptoms. And by and large, those long COVID symptoms just span the range from neurological to fatigue and respiratory symptoms. Females are more likely to develop long COVID than males, but certainly long COVID is at another level, an equal opportunity employer. One thing that we looked at, and I think this probably resonates with people that have some of these chronic conditions, is that we wanted to help define the condition. In other words, in many of the patients that we've interacted with, they have a great deal of frustration with the medical system because they'll go in, they know things are wrong, and yet there's not a clean diagnosis in which the physician says that they agree with the patient. And so we looked in our patients at proteins or metabolites, the kind of things that can normally be used as disease markers to identify irregularities in health. And we found two things. The first is that we found that patients that had neurological sequelae, meaning brain fog, things like this, we could identify very little in their blood that correlated with that. But what we did find was very significant, and that was that they had a disrupted circadian rhythm that we could quantitate through the measurement of 2 proteins out of something like 500 that we measured. And then for patients that had sort of respiratory viral-like symptoms, meaning flu-like symptoms, fatigue, et cetera, we found that those patients at recovery had very low cortisol or cortisone levels. And you can see that there's a lot of stars here. That means that's significant compared to patients that don't have those symptoms or patients that are healthy. And this recalls a condition called Addison syndrome, or when it's very severe, Addisonian crisis. And it has to do with what's called adrenal insufficiency that has a very common set of symptoms as long COVID. Oftentimes this can be treated by cortisol replacement therapy. Physicians may wonder if we're seeing low cortisol because of steroid treatments that these patients often receive. Many patients with COVID-19 will get steroids, and for sure, at initial disease, a diagnosis or an acute disease, and you see these big pink things show that in fact, there is a strong correlation of low cortisol with steroid treatment, but at recovery, there's no correlation whatsoever. This seems to be intrinsic to these patients that have these sort of respiratory viral-like symptoms. And so this seems to be something that probably may be treatable, and I think there is some physicians are beginning to use cortisol replacement therapy in treatment of patients. Another thing that we looked at is the reactivation of latent viruses. So, it's bad enough to get COVID-19, but there were some indications in the literature that COVID-19 may actually wake up viruses that are in a latent state in the patients. And two viruses for which that could be problematic are Epstein-Barr virus, most commonly associated with mononucleosis, and cytomegalovirus. And so most of you all on this call are going to have, at some point or another, been infected with EBV. You may not have had mono, but doesn't mean that you don't have EBV latent in your body. And about half of us have a CMV, and these is not active. It's sitting in some reservoir in your body in quiet stage. And so when you reactivate a latent virus, there's two ways to see it. One is to look for antibodies against that virus, but those antibodies will last for a long time. And so it's very difficult to understand when you look at antibodies if their reactivation was associated with COVID-19 or with something else. Or you can look directly in the blood for genetic signatures of the virus, and that's what we did. And that's very transient. So it's associated with a particular event. It only lasts a couple of days. And so when we did that in our patients, we found no evidence of CMV reactivation, but we found in about 20% of our patients that Epstein-Barr virus was reactivated. And you can see that this is the blue lines here, and it's almost not detectable by the time the patients have recovered, although you can see it in these patients right at diagnosis. And similarly, we found that SARS-CoV-2 virus, the genetic signatures of that virus, which is, you see that through RNA, is also present in a subset of patients. This is mostly associated with disease severity. EBV reactivation is not. And in fact, you can see that this is associated with disease severity because over here, the patients that had high levels of SARS-CoV-2 RNA in the blood were more likely to succumb to the disease. So when we look at these two viral signatures in patients that recovered and are convalescing, and here we've corrected for disease severity, what we find is that these viral signatures, by and large, associate with neurological long COVID. Things like memory problems and difficulty concentrating, things like this. And I think it's interesting that simultaneously with our paper being published, another paper was published that was associating Epstein-Barr virus mononucleosis associated infection in young people with the development of multiple sclerosis later in life. So I don't know if there's a relationship between those two findings, but it is intriguing and perhaps a little alarming that these reactivated EBV virus associates with neurological PASC. Autoantibodies. I think there's been a lot of press on autoantibodies and how they can anticipate perhaps a fatal COVID-19 infection, especially in older individuals. And in fact, the ones that are most important are autoantibodies associated with what's called type 1 interferon or interferon alpha in this case. And so we looked for autoantibodies in these patients. We also looked for another set of autoantibodies associated with lupus or systemic lupus erythematosus. And the reason why we did that is if you dig through the literature, the scientific literature on lupus, you'll find that there's a particular type of B cell. B cells are what make antibodies. There's a particular type of B cell that matures in an unusual way. It's called an atypical memory B cell, and it's associated with lupus. And in a paper that we published about 1.5 years ago in Cell, we had identified the presence of those types of B cells, this unusual type of B cell called atypical memory B cells. And that made us think that maybe there was a connection between long COVID and lupus. And because of that, we measured a host of autoantibodies that make up a common panel when one's looking for autoantibodies associated with lupus. Okay, so what did we find? So here is this complicated looking plot. Down here are the autoantibodies, and up here are different variants of the antibodies that are protective against the coronavirus. Number one, we found that if you have one autoantibody, the fact that all this stuff around here is red says that you're more likely to have other autoantibodies. Number two, if you have autoantibodies, they repress the levels of mature protective antibodies against the virus. In other words, patients with autoantibodies may be more likely to have serious breakthrough infections because they have a little less protection against the virus. And so the possibility for reinfection is higher. Third, when we looked at how these autoantibodies associated with different respiratory and neurological, et cetera, different long COVID symptoms, we found that they associated with sputum, meaning people are coughing up stuff, gastrointestinal distress, or inability to exercise. And so this also suggests that autoantibodies are associated with long COVID. More importantly, we found that patients that have autoantibodies at recovery pretty much had autoantibodies at initial diagnosis, and that those autoantibodies were mature. And what that means is that these autoantibodies predate the infection. That may not be true for all patients, but it's true for the majority of patients. Now, the autoantibodies we're reporting here, something like 20% of our patients had autoantibodies. That's a much higher number than if you went into the clinic and had, say, a test done by LabCorp. And that's because LabCorp is going to present is going to tell you positive for autoantibodies that they're at what's called a clinical level, meaning at a level that is commonly going to give you symptoms. A larger fraction of patients will have subclinical levels of autoantibodies. We find those are actually important for long COVID, even though most patients would not have known that they had them. What we also found is that for the patients that had very high levels of autoantibodies, meaning at a clinical level, there were immunological signatures such as these higher levels of atypical memory B cells in these patients. And so these autoantibodies absolutely affected the disease journey a patient takes through COVID-19 infection, but they also are what we call PASC factors. They anticipate long COVID. I hope that makes sense. It's a little bit complicated, but I think it's an important concept. Now, we only measured this limited panel of autoantibodies. At the moment, we're looking at autoantibodies against the entire human proteome to understand the extent to which these autoantibodies are important. But the other thing that this establishes is that there, at some level, is a relationship between some patients suffering from long COVID and SLE or lupus. And that's an avenue that we're very aggressively pursuing now. And with luck, it may lead to potential treatments. So we did a lot of work on single cell biology, and I'm gonna just give you a little bit of insight into that, because it told us that one of the long COVID symptoms that patients develop is really quite unique. So when you have, this is kind of a lot of jargon here, so you can ignore the jargon. I'll just try to describe it in plain English. Before you have an infection, you'll have T cells circulating in your body that are with great diversity, meaning that you have the ability, the T cells have a diversity such that almost anything that your body sees that's, say, a foreign pathogen, at least one or two T cells has the capacity to recognize that foreign pathogen, like coronavirus. When you get exposure, those selected T cells that can recognize a virus will expand. This is why your lymph nodes get bigger, but you have a lot more T cells because they go on the hunt and they can kill cells that are infected with the virus. As you recover, these T cells should contract back and just form protective memory, okay? And that generally is what happens. But we can use these T cell, the unique genetic information in these that tell us what these T cells recognize as barcodes to follow T cells through the course of disease. And so we did that. We used the T cells as barcodes, and we followed these patients. And what we found, this is a technical plot, but I'll describe it in simple terms. We found that patients that develop gastrointestinal long COVID, gastrointestinal symptoms, did not behave as they should have. In other words, their T cells were still aggressively expanding at the time those patients were recovered from the acute symptoms of COVID. And on top of that, T cells against other viruses were also aggressively expanding in those patients. And so it's a non-specific immune response, but it's basically chronic inflammation that is in the T cell compartment that is associated with gastrointestinal long COVID. Probably is because that's a late developing a symptom compared to some of the other ones. And so this is not one that we can anticipate at diagnosis. That actually evolves more slowly, but it's one that you can anticipate just a few days after diagnosis. Okay, so finally, we put all of this data together and we tried to understand, are these different factors that we see that anticipate long COVID, how are they related? So how might a physician think about them? Is there one drug that can cover all these different factors, or is there going to be many, many drugs? And what we found was very interesting, and I illustrate it here. So in this plot, what I've shown are the relationships between all these different autoantibodies, type 2 diabetes, the different viral measurements that we made of Epstein-Barr virus and SARS-CoV-2. And you see there's a fair amount of color in this graph. And this suggests that some of these past factors are somewhat dependent upon each other, and that there's perhaps a limited number of therapies that could be used to address the impact of these past factors. This is at diagnosis. If I look at a week later, I find that a lot of this color has disappeared, but there's still some there. But I look out at 2 to 3 months later, I see no relationships at all. And so this is what oftentimes physicians see when you go to them. You've got these vague set of symptoms, and it's crazy hard to track back where did these symptoms come from, which is one of the challenges with this and other chronic diseases. And the issue is that your body just scrambles that information over time. It's actually there if you can capture it when the infection happens and that triggering event that leads ultimately to long COVID out here. But once you have the long COVID, very, very difficult to go back in time and understand what caused it. And so I think this is, in a nutshell, why some of these chronic diseases are so very, very hard to treat. And so, so let me just summarize a little bit. So each of these factors, EBV viremia, so reactivation of Epstein-Barr virus or the SARS-CoV-2 viral loads in the blood, account for about a third of the long COVID that we found, as does type 2 diabetes. The autoantibodies accounted for about 2/3. And so the reason why that 1/3, 1/3, 1/3 plus 2/3 adds up to more than 100% is some patients had both. But basically, the autoantibodies are the most important. But these three are also important, just at about half that level. So a word of caution. Most of these autoantibody levels would not be reported back by standard clinical labs. They're at subclinical levels, but doesn't mean that they're not important. They actually are important for long COVID. They're just at lower levels than what would be come back in a clinical lab. Patients that have autoantibodies at very high levels, for sure it's a stronger predictor, but nevertheless both of them will predict. And these high autoantibody levels not only can anticipate long COVID development, but they also may be associated with susceptibility for reinfection. A few limitations. So we only went out 2 to 3 months post-diagnosis. There's a big national study right now called RECOVER. I'm leading the Pacific Northwest branch of that, which involves many hospitals up here. And we'll be looking out at patients out to 4 years post-diagnosis. So we can't say about very long-term effects yet, but that's just a caveat of what I just talked about. We only saw about 300 patients in our study. So some of the rarer conditions like hair loss or cardiac issues, we probably don't have the statistics to address. It doesn't mean they aren't real. We just don't have statistics to say much about them yet. The reason why we resolved type 2 diabetes is because we do have those statistics. We only looked at a handful of autoantibodies, and so now we're looking at many more. And this may allow us to make other associations with other autoimmune diseases, such as the one we made with lupus. For sure, long COVID is going to have some dependence upon what genetic variant of the virus you were infected with. And most of our patients were pre-Omicron. The ones we're studying now are not. They're probably include everybody, all the different genetic variants so far. But we don't know how Omicron is going to differ from, say, Delta and pre-Delta variants. And then, of course, this long study, the national study that we're a part of, will look at many patients. So we'll answer many of these limitations. And I want to just make a plea that, we couldn't have done this study without patients consenting and complying and, really giving up their time and their blood to help make this happen. We're absolutely grateful that they did that. And it's going to take that again in spades for this to really resolve what this disease is about. So for those of you who have participated in studies, we're very grateful to you, and hopefully you'll continue to do that. And finally, we received these are not cheap studies to run. We received funding from a number of foundations and federal sources, the NIH, BARDA, et cetera, that we're grateful for. Thank you.
Thank you. Oh my goodness, what great information. And I love what you said about the willingness of individuals to participate in studies, because that's how we learn, and that's how people get help. Thank you. Thank you for your research. Thank you for that wonderful information that you shared. And at this time, I would like to have our sponsors, CSL Behring, Grifols, Takeda, Accredo, and ADMA Biologics. They are going to share some videos with us about the impact that they make in the PI community. So here we go.
For more than 100 years, we've turned innovative thinking into solutions. CSL's investment in innovation has helped to develop a robust pipeline of safe and effective medicines and improve existing ones so that we can bring patients meaningful innovations and advancements that make their lives better.
Hi, my name is Ricky Mensch, and I'm one of Grifols immunoglobulin certified nurse educators. As part of Grifols, whose mission is to improve the health and well-being of people around the world, the nurses support the PI community through education of healthcare providers on all aspects of IG care. Our goal is to improve medical outcomes and quality of life. Grifols is dedicated to ensuring that people who need plasma products have access to them. They do this through plasma donation awareness, sustainable supply efforts, and education to increase the early identification and treatment of PIDD. For more information about Grifols, please visit us at grifols.com.
My name is Dana Fladhammer. At Takeda, I am the Director of IG Community Support. You might know the team I'm lucky enough to be a part of as the MyIG Source Patient Advocates. I know that throughout my journey as a mom of 4 children living with primary immunodeficiency, the IDF has been simply an invaluable resource to me. I hope you learn a ton, and if you need us, know that nobody should ever have to do this alone.
Welcome to the IDF Forum. I am Kim Farrell, Product Line Manager with Accredo. Accredo is a best-in-class specialty pharmacy with a nationwide network of immune globulin-trained home infusion nurses and pharmacists. It is truly an honor and privilege to be part of this immune deficiency community. Accredo places patients at the center of all we do. We deliver individualized care and support for patients living with immune disorders. It's all about giving you a great and personalized pharmacy experience.
And on to the Q&A. So at this time, I would like to invite my colleague, Colleen Brock, Manager of Medical Programs at IDF, to moderate the Q&A session. And I will now turn it over to you, Colleen. So please take it away.
Hello, Kathy, Dr. Heath, and everybody here this evening. That was a wonderful presentation, and we have a ton of questions. So let's get started. So do you know if there is any incident in the percentage of people who are developing long COVID after having a breakthrough infection? So they've been vaccinated, they got sick. Are their chances of getting long COVID greater, lesser, the same?
My guess, so I don't know if the epidemiology on that has been done yet. I would hazard a well-educated guess that the chance of getting long COVID is greatly reduced by being vaccinated and boosted. Basically, you're keeping those viral levels down low. You already have an immune response that is at least somewhat responsive to the virus. And so it's going to have a smaller chance of allowing for the types of past factors that I talked about, 2 of which are viral signatures, to impact the long-term recovery.
So if a person is diagnosed, what treatment options are available?
It's a good question. So the primary treatment option that would be suggested by our findings would be some of these antivirals that are coming out now. I think Merck and Pfizer both have them. If you'll recall, when we measured those viral loads in the blood, they were there very transiently. And so the implication is that the antivirals need to be used as early in the disease course as possible. There's a caveat there because the two viruses that we found in the blood, SARS-CoV-2 and EBV, one is an RNA virus, SARS-CoV-2, the other is a DNA virus. There's actually different antivirals. But that would be the strong suggestion right now would be that you do that. We know that these autoantibodies are functional, meaning that higher levels of autoantibodies absolutely impact a patient's course through the disease and recovery. That suggests that there are other druggable targets that are relevant. And I think we can, if you look at this long COVID as a way of studying the emergence of autoimmune diseases, COVID is just a perturbation. It's really done amazing things for allowing us to begin understanding some of these, like lupus. I talked about lupus. And I'm sure we'll find others. And those are challenging to develop treatments for many reasons. One is that we don't actually have a triggering event. And so you're always looking after the fact when things are somewhat randomized. Now we have a triggering event. We actually see the time course. We actually know in some ways when the lupus-like symptoms originated. But also I had 300 patients give their time and blood. It gives us the statistics to begin resolving things like what happens when you have really high autoantibodies or what the connection to lupus. And that's how you can develop treatments. So we're just beginning to have treatments, but it's a big deal to have the numbers of patients that we had to begin resolving some of these connections.
Okay, thank you. So in the PI community, so those with antibody deficiencies and others, do we have any idea what the complications are for those people that get COVID?
Yes, we do, for at least some of those conditions. So we have studied a number of patients with, say, MS and patients with that had serious immunodeficiencies, that organ transplant patients. And MS patients actually will eventually, depending on the therapy they're on, they will eventually develop an immunity against the disease with vaccination and boosting. It takes quite a bit, more than the average person, but they eventually do develop responses. In the absence of that, the T cell responses they develop do lend some protection. So even though they don't have much in the way of B cells, the T cells can lend some protection. It's very dependent upon the therapy that you're taking. For seriously immunosuppressed patients, you don't have much of a defense against the disease, and the monoclonal antibodies are about the best way to treat patients if they do unfortunately get infected with COVID-19.
Okay. Do you know if the severity of long COVID is determined by what immune disorder you have?
Almost surely.
Okay.
I don't think we have that fully resolved yet, but I did point out that if you have clinical levels of autoantibodies, there are stronger indicators of long COVID than subclinical levels. They both can be indicators, but that just means more autoantibodies is not as good for the patient. How that parses between different autoimmune conditions, for example, I don't think we have the statistics to resolve yet, but it's almost certainly going to be important.
You had talked about type 2 diabetes being a high risk factor. Why not type 1?
That's only because we had 40 or 50 patients with type 2. Type 1 is much more rare. And so we didn't have the numbers to resolve the impact of type 1 diabetes on long COVID. It could well exist.
Can long-term COVID be diagnosed 2 years after your original infection?
I'm guessing that we're going to find out. The answer to that is probably. it's going to be challenging to know if it's really associated with COVID, but your body is going to have in it the immunological history of your COVID infection because you have antibodies. And it's going to have the immunological history of whatever autoimmune signature you might have that could be a long COVID symptom. And so it may be possible eventually to read those two out and understand they're related. But I think, 2 years out, that's a challenge now. We have a hard time going back a couple weeks, but and understanding what happened a few weeks ago. So understanding what happened 2 weeks ago it's not out of the question, but that would be state-of-the-art science to try to do that.
Do you know if any of the patients you studied had tinnitus from COVID?
Oh, I don't know. If we did, it wasn't enough patients to say much about.
Now, in the study that you are conducting, the one that has the 17,000 patients, are you going to have patients in there with primary immune disorders such as CVID or DiGeorge or anything like that? And if so, how do people get in touch with you to be involved with it, or can they?
So this is a great question. And in fact, we are, I just had discussions with physicians today about making sure that we had sufficient controls for exactly those types of conditions to compare against patients that are coming in with long COVID, because we think there's going to be a lot of parallels. I think if you, for those controls, we'll probably have our own local study, which we will try to recruit patients, and we'll advertise that when we're doing that. For the National Recover Study, I think if you Google the National Recover Study, you will find an opportunity to participate, and your email, they'll ask you probably for your zip code or something like that. And then if you qualify to be in a region, there's something like 15 different sites that are spearheading recruitment. You will be directed towards one of those regions. They're trying very hard to sample the broad demographics of the US. I can tell that's a major goal that we're trying to do here in the Pacific Northwest. And so there may not be a site near you, but I think the odds are pretty high that there may be.
You had talked about cognitive issues. What can be done to help those people?
That's a really good question. And I don't think we have good solutions yet. I can tell you that in most patients, the cognitive issues will dissipate over time, but whether there's an effective treatment that can help those patients, I don't think we have enough information about the nature of that cognitive dysfunction to know.
What about in situations like fatigue or GI symptoms?
Well, some of the, like fatigue, I think at least some patients, this cortisol replacement therapy may be a viable option. For GI, I don't know. I think I'm not a physician, so I don't want to give too much physician advice here. I would say that these long COVID clinics are trying a number of things, and there's a number of companies now, because of the magnitude of this issue, that are beginning to trial drugs. They'll be trialed through this Recover program as well as other programs. So if you are interested in some of these drug trials, probably the Recover program is the place where they'll be tested.
So if somebody who got COVID and was not vaccinated, but they got prophylactic monoclonal antibodies such as Evusheld, are they less likely or more likely to have long COVID?
I think they're probably about the same, would be my educated guess. I don't know, it'd be about the same.
What is the impact of being on multiple immune suppressants as well as having a primary immune disorder in the risk of long COVID?
It's very immune suppressant dependent. So I can't answer that, and I don't think anyone really knows. But I think the chance of it, of having COVID, being on immune suppressants, and getting long COVID, is significantly higher, because your viral load is going to go higher, and all the various factors I discussed are probably going to be present. You're going to have less of a B cell response, things like this. But it's going to be very dependent upon what therapeutic regimen you are on. And I don't think there's not enough data yet and not enough epidemiological studies to know which drugs, which immunosuppressants are associated with which long COVID symptoms and which ones are better.
So if you have an immune disorder where you don't produce B cells, and if you do, they don't work very well, but you have T cells that function, what are the odds of protection against long COVID in that situation?
I don't know. I do know these are all really complicated questions, and it's early days yet.
Yeah.
Because this, as you can imagine, the science I told you, we basically worked night and day, and it was not overnight to get this out. I can tell you that if you do have functional T cells, your capacity to develop immunity against COVID is reasonable. It's not great, it's reasonable. The T cells do respond, and they can be effective. How that parlays into long COVID, I think, is anyone's guess. My guess is its odds are higher that you'll have some sort of long COVID symptoms. They may take longer to dissipate, but I don't think anyone knows yet.
So is it true it takes immunodeficient people longer to clear the virus, and that we are responsible for creating some of these variants that has kind of circulated through the news, that variants are caused by immune deficient patients who are harboring the virus?
It's very specific.
Case, how do I protect somebody else from getting it for me if I still have it in my system?
That's a great question. It's very specific individuals that are immune deficient, but that is true. Not just all immune deficient people that are going to promote new variants of the virus. It's very specific ones. I don't think we fully understand who those are, but we can if you do the right measurements, you can identify someone who does qualify. And I think the best way is just if you are immunodeficient and you have COVID-19, to isolate until you basically are recovered. And it's really important to isolate until you're recovered, because there is a chance that you're making new variants.
Yes. Have you seen any result with patients that have lymphoma T cell and severe COVID?
I've seen patients with leukemia, certain leukemias, and other cancers in COVID. There is the effect of immunosuppressors that we've already discussed about how much we do or don't know about them. There's only one leukemia that really led to much more severe fatalities than non-cancer patients. I think it was CML, but I'm not sure. I can't remember. But whether these patients are going to have long COVID with higher regularity, I think is unknown. We've been working with Sloan Kettering a little bit to try to resolve some of these issues, and there's a number of studies going on, but it's just not known yet.
What effects do lack or very low numbers of memory B cells have in your risk or recovery?
Well, I think if you're talking about for reinfection, that's one of the motivations for boosting. In my estimation, at least for young people, even if you don't measure much in the way of antibodies, if you've been vaccinated and you look at antibody titers in a patient, there's not much. If you get reinfected, your antibodies crank up really, really fast. So I think boosting is not that important for younger people, even if they have relatively low antibody titers. Now, low B cell numbers is different, but low antibody titers. For older people, that actually is quite important. But if you got low B cell titers, that may indicate that you've got an autoimmune condition, and then all the other complicated things that we've not been able to very effectively address so far are probably important.
Yeah, there's a lot we still don't know.
Absolutely. Most of this we don't know.
No.
It's amazing how much we do know at this,
It is
with
in a
only a
short
couple
period of
years
time.
into it. Yeah.
You researchers are the backbone of our knowledge.
Yeah, it's been, we're trying.
Well, we appreciate it.
These are hard questions though.
They are. This is a very smart audience. Somebody wrote, a recent study published revealed that COVID-19 infection can set off massive inflammation in the body. How does that affect a person with PI? And I also had another question about how is it related, or how does it affect somebody with mast cell?
Yeah, mast cell, I'm not going to be able to tell you. I'm sure there's an effect, but I don't know what it would be. For high levels of inflammation, there were a number of drugs that were, in fact, the steroids were largely used to try to control that, but also things like tocilizumab, which was an IL-6 receptor inhibitor to suppress out of controlled inflammation. I think this has become, the biggest issue here is when you've got older patients that either have dysregulated high inflammation responses that can get out of control. And for sure, that has a huge impact on acute disease. It probably impacts recovery because, if you've had more severe disease, you're going to take a longer time to recover. But how it impacts some of these emergent chronic symptoms, I don't know. I think that's an open question.
So what testing can a doctor do to see if you have autoantibodies? And if the patient is post-COVID and still having issues, can existing autoantibodies inform the treatment? So how do you test
existing autoantibodies? Existing autoantibodies, you can test for them using, there's LabCorp-like assays that are done, that are commonly done for lupus and things like this. They're not crazy sensitive. In fact, as I tried to indicate, they report back clinical levels of autoantibodies, not subclinical. But at least if you have really high levels, you'll find that out. So that's a standard clinical test. I'm hoping that more sensitive variants will come out, but you don't want to overtreat patients. So there's a balance there that you have to strike. And the other question is, what does that mean in terms of treatments? I think it means a couple of things. Right now, I can tell you there's a lot of active epidemiological work going on, trying to understand for certain autoantibody signatures and certain treatments that have been used for, say, lupus and things like this, what has been most effective? And are those treatments relevant to treating long COVID symptoms? The other issue is that, as I also tried to indicate, when you have high levels of autoantibodies, we can actually see that they impact your immune function. I mean, they're functional. And that suggests that they do constitute, at some level, a drug target, whether it's directly those autoantibodies or something in the chain that produces them. So that's an active area of research. I'm hoping that that's an area that can be accelerated as we begin trying to, hurry up and find a way to treat these patients. But I don't know if there's a treatment right now that would be effective for specific autoantibodies. I think that's just unknown.
So in the studies that you're talking about, and that you're doing, are you separating those who have been vaccinated from those who have not?
We are recording all that information.
Okay.
And so at the end, with 17,000 patients across the nation, you'll have patients with the whole spectrum, and that's all information that will be used to interpret the findings. You basically biobank and record everything and clinically characterize the patients as fully as you can, as well as do some of the types of sort of molecular measurements I talked about. And then you let you just let the data sort it out, with,
I'm going to take you a step back in that, so our audience understands, when you're talking about autoantibodies, what is the difference between autoantibodies and antibodies in their system?
So antibodies are molecules, or big fat molecules shaped like a Y, that are produced by your B cells against something foreign to your body. And so, for example, the coronavirus vaccines are designed to produce antibodies against the spike protein, in fact, the receptor binding domain of the spike protein, okay? And so that is a process that takes, you give the vaccine, and then the B cells and T cells have to talk to each other and mature. It takes a few weeks. So that's why you get a vaccine, and then a certain amount of time later you get the booster, but it takes a while to get immunity. Autoantibodies are basically when your body is making exactly those same kind of molecules, but against proteins that your body naturally makes. And so autoantibodies can be against, for example, I talked about anti-interferon, class 1 anti-interferon alpha. Interferon alpha is actually an important signaling molecule in your immune response cascade. So when you have autoantibodies that are hitting interferon alpha, they're blocking the function of that important signaling molecule. That's how it affects that's an unnatural thing. Your body shouldn't be doing that. That's how it affects your disease journey. But there are other autoantibodies that can be against proteins that are inside cells. So proteins that normally aren't exposed to your immune system, but when you have cells dying, those proteins do get exposed. And many lupus-like autoantibodies are those kind of autoantibodies, and so on and so forth. So different disease conditions will have different autoantibodies, and they, proteins are little machines. They're going to impact the proteins in ways that are specific to that disease or to that patient.
So somebody who has rheumatoid arthritis, somebody who has other diseases like that are dealing with autoantibodies. Now, I just lost my train of thought. All right, we'll just move on to another question and it'll come back to me. Oh, so if you have COVID and you've had it, you had all the symptoms, whatever variant you had, is it possible to create autoantibodies to COVID in your body?
No, those by definition would be antibodies.
Okay.
Because COVID's a foreign entity. And so, so any antibody against COVID is going to be an antibody, not an autoantibody. Auto is to self.
Right. Is the COVID virus, is it a latent, can you have it as a latent virus that is reactivated, kind of like EBV and CMV, at a later point in your life?
That is a great question. And the answer is a solid maybe. So for it to be latent, it has to basically have a tissue reservoir where it sits and basically hides out and just is quiet. And there's some recent work on autopsies that suggests that there may be such reservoirs, but we don't really, you've got to do a lot of patients to know if that was a one-off identification or if that's actually a real thing.
So there are a lot of people here this evening who either have a primary immune deficiency, a secondary immune deficiency, have other autoimmune disorders. We are a wide variety of things. So if we are suffering from long COVID, irregardless of what the symptom is, how and can we be involved in a study? And also, where do we find a clinic with a physician, the researchers, the studies who are going to understand when we come in and say, I have fatigue, I've got a cough that won't go away, I've had diarrhea for 3 months now, and I've lost 30 pounds. Where do we go?
That's a good question. If you can go to one of the clinics that is participating in the RECOVER study, you're likely to find at least a sympathetic physician. That doesn't mean that they're going to be able to do much about your treatment at this stage, but you're likely to find someone who actually understands what's going on. I know most health systems have set up some sort of long COVID clinic many places. And it may have to travel a little bit to go. That would be just the healthcare system. It's not going to be involved with RECOVER, but there are a number of long COVID clinics. We work with several here in town, but I would say also, I mean, this is, I indicated this earlier in my talk when I talked about that we measured these like disrupted circadian cycle and stuff. We're just trying to define the disease so that you have a measurement that says this is something's really wrong here. I think there is a growing appreciation that is the case. There's probably a growing frustration to accompany it because we don't really know how to treat those patients yet. But that's putting pressure on researchers like myself to try to identify treatments.
So the patients that you studied, do you have any idea yet? I know you said you only studied them up to the 4-month period, but do you have any idea how long it takes to get over some of the symptoms and which is the hardest to overcome?
Yeah, we have some patients that we've studied out to 6 months or even a year. At let's say 2 to 3 months, in our patient population, almost 40% of the patients had some sort of chronic looking symptom. By 6 months, it was down to 15%. And then after that, I think it decays a little more slow.
All right.
But that does tell you that most patients that are feeling like miserable after 2 or 3 months post-infection will actually be okay after 6 months. But there is a non-trivial number. I mean, 15% is pretty good sized number that are still going to be feeling something 6 months out. And, maybe, only a third of those are be recovered a year out. I don't think we know all these numbers yet, but that's kind of our educated estimates on.
What was the most significant symptom that you saw that lasted the longest?
It's a good question. I think it would probably be fatigue or respiratory issues, but I'm not sure. I could be wrong. And maybe even for some patients, some of the neurological ones. But I think once you get out to a year, the symptoms are a little more uniformly spread than they are at 2 to 3 months, where respiratory and fatigue issues are more important.
Okay.
And then there could be symptoms that emerge that we simply haven't seen by 2 or 3 months out, but cardiac issues that may be subclinical at 2 or 3 months may actually become clinical. That's the thing that people are very concerned about right now.
Yes, I think we have a lot to learn.
Yeah.
And like I said, we can't thank you enough for all that you have provided tonight, all that you will continue to provide. We hope maybe you will come back in the future and tell us more that you've learned since you're working so many hours, but we can't thank you. And I'm sure you have an entire team behind you that are working night and day to help everybody that's still struggling after being sick.
I do. It's a great team. And they were, in spite of the fact that those questions were really, really hard, it was really great questions. And I'm glad the audience was so engaged and interested. Thank you.
Thank you. They are a very smart group, and we truly appreciate your time this evening.
My pleasure.
And Kathy,
Bye-bye.
I'm going to hand it back to you.
Oh my goodness, Dr. Heath, we have wonderful presentation and wonderful comments coming in, and I will be sharing those with you in the upcoming days. So again, thank you. And thank you tonight to our featured sponsors, CSL Behring, Grifols, Takeda, Accredo, ADMA Biologics, X4 Pharmaceuticals. And thank you to each and every one of you who attended, participated in tonight's forum, because your questions make a difference and they make these forums as good as they can be. So you are amazing. Thank you so much. And with that, we would like to wish you a wonderful rest of the evening and enjoy, take care. Please remember that IDF is here with you every step of the way on this journey. And we look forward to seeing you next time. Good night.